This is Lauren Clifton. I'm in Anatomy and Physiology with Professor Langford, and I'm doing my lab video for my extra credit. We're going to do it over mitosis. I'm going to show you the first two steps of mitosis. We're going to start with interphase. Interphase is a period of a cell's life when it carries out its normal metabolic activities and grows. Interphase is not part of mitosis. During interphase, the DNA containing material is in the form of chromatin. The nuclear envelope and one or more nuclei are intact and visible. There are three distinct periods of interphase. G1 is when the centrioles begin replicating. S is when DNA is replicated. G2, the final preparations for mitosis are completed and centrioles finish replicating. Okay, in early prophase, the chromatin condenses, forming bar-like chromosomes. Each duplicated chromosome consists of two identical threads called sister chromatids, held together at the centromere. Later, when the chromatids separate, each will become a new chromosome. As the chromosomes appear, the nuclei disappear, and the two centrosomes separate from one another. The centrosomes act as focal points for growth of a microtubule, a symbol called the mitotic spindle. As the microtubules lengthen, they propel the centrosomes towards opposite ends or poles of the cell. Microtubule arrays called asters or stars extend from the centromere matrix. Late prophase. In late prophase, the nuclear envelope breaks up, allowing the spindle to interact with the chromosomes. Some of the growing spindle microtubules attach to kinetochores, special protein structures at each chromosome's centromere. Such microtubules are called kinetochore microtubules. The remaining spindle microtubules that are not attached to any chromosomes are called polar microtubules. The microtubules slide past each other, forcing the poles apart. The kinetochore microtubules pull on each chromosome from both poles in a tug-of-war that ultimately draws the chromosomes to the center or equator of the cell. I'm going to pass the phone to Hailee.
Alright, I am Hailee Fenley, and I'm doing the video for extra credit, obviously, for Mr. Langford as well. I'm in 2401.081. And these are the last three steps of mitosis. Alright, metaphase. The second phase of mitosis. The two centrosomes are at opposite poles of the cell. The chromosomes cluster at the midline of the cell with their centromeres precisely aligned at the equator of the spindle. This imaginary plane midway between the poles is called the metaphase plate. Enzymes act to separate the chromatids from each other. Anaphase. Third phase of mitosis. The shortest phase of mitosis, anaphase, begins abruptly as the centromeres of the chromosomes split simultaneously. Each chromatid now becomes a chromosome in its own right. The kinetochore microtubules moved along by motor proteins in the kinetochores gradually pull each chromosome toward the pole it faces. At the same time, the polar microtubules slide past each other, lengthen and pushing the two poles of the cell apart. The moving chromosomes look V-shaped. The centromeres lead the way and the chromosomal arms dangle behind them. Moving and separating the chromosomes is helped by the fact that the chromosomes are short, compact bodies. Diffuse threads of chromatin would trail, tangle, and break, resulting in imprecise paracelling out to the daughter cells. The final phase is telophase. Telophase begins as soon as chromosomal movement stops. The final phase is like prophase in reverse. The identical sets of chromosomes at the opposite poles of the cell uncool and resume their thread-like chromatin form. A new nuclear envelope forms around each chromatin mass. Nucleoli reappear within the nuclei and spindle breaks down and disappears. Mitosis is now ended. The cell for just a brief period is binucleate, has 2 nuclei, and each new nucleus is identical to the original mother nucleus. Cytokinesis is a division of cytoplasm. Cytokinesis begins during late anaphase and continues through and beyond telophase. A contractile ring of actin microfilaments forms the cleavage furrow and pinches the cell apart.
Okay, I'm going to go through and label the parts of the cell as they're going through the different steps of mitosis. So in interphase here you can see the centrosomes, and each one of those has 2 centrioles. And then this around here is the plasma membrane. Inside of the nucleus we have the chromatin, and around the nucleus is the nuclear envelope, and then here is the nucleolus. Okay, so when you go to early prophase, this is when you have the early mitotic spindle, which is up here. And then now you have the centromere, which is right there in the middle. And then you have the chromosomes, which are consisting of 2 sister chromatids. And then over here in late prophase, you have the spindle poles, the kinetochores, which are in the middle, polar microtubules, which are those that do not have the kinetochores. And then you have fragments of the nuclear envelope, because this is when the nucleus breaks apart. And then you have right here the kinetochore microtubules. Okay, and then moving on to mitosis. Right here is the metaphase plate, or the equator right here across. And you can see it here in an actual picture. And then you have the spindle, which is this whole thing here. In anaphase, we have the daughter chromosomes as they split apart. You'll also see that called sister chromosomes. Okay, and in telophase and cytokinesis is when the cells actually begin dividing. Right here at the cleavage furrow, you see that how it's actually splitting into 2 cells. And then you have the nucleolus, which is actually reforming, and then also the nuclear envelope, which is reforming, and you can see that on both sides. Alright, well, there you guys have it. That's mitosis.